I'm here. Can you hear me?
Yes, we can hear you. How about you share your screen and start?
Let's see.
Thank you.
Screen. Ding. You see my screen pop up?
Yes, all as well.
All right. All right. So I hope it moves okay, but if the animation looks a little bit weird, I'm sorry about that. I'll fix it if it needs to. So the screen resolution seems to change a bit. Hi everyone. My name is Ice. The name is hard to pronounce. It's Thai names, so just ignore it and just call me Ice, and you see me through the course as part of the TA team. So the talk I would like to share with you guys here is to work around the colored proteins so that we can use it for some of the BioArt application that we're gonna have here in this year of How to Grow Almost Anything course. What I show you in the title slide here is how to turn some of the colored protein into some kind of art. So you can see that this strawberry is going to be pretty hard to draw on if you have only one color. But in the case where you have multiple colors in the multiple shades, it's possible to turn it into this kind of beautiful artwork. So before we go into the deeper story about how we make these kind of artwork, I wanted to share to you on how these works can be worked around using the basics of the biochemistry and some of the genetic engineering we had. So this can be connected to a lot of works that people has been talking today. So to get started, I hope this slide goes well. Yeah. I want to show you first, how these colors in nature sort of form. So this is, some images that I got when you just try to search around the colors in nature. You can see that, a lot of colors were already available in the nature, and most of these colors were formed for different purposes. So you can see that the shades of yellow and the shades of red were kind of different, and you see a lot of these different shades as we move forward through the course as well. So if we go a little bit deeper, you know, for some of you who live in the geographics where you can have different seasons, you see these kind of different shades in the autumn or in the fall, and they are composed of different kind of pigments. So how do these pigments form and how do the nature sort of control how these were made is dependent on different kind of, biomolecular compounds or the structure by itself. So I just want to clarify here that most of the things that you've seen on the left-hand side on these, different shades of leaves, of autumn leaves, were based on the compound itself. But if you go to some kind of, colorful, vibrant colors on the right-hand side is actually from the structure itself. So these are not compounds. It was like the butterfly or the birds were having like some critical structure in their feathers or in the wings so that they can behave similar to how you can see these bubbles here. And you can program them pretty easily if it's a compound, but it's going to be a lot harder if you have to make these microstructures that it can reflect the light. So in this right hand side is the process called iridescence, and we're not going to go too far on that one. I just want to make sure that there's some colors that may not be as easy for us to modify or program in biology. We're not going to go too far on that, but these are some examples of the bacteria that can be engineered so that they can have these kind of colors, similar to how the birds or the butterfly can have as well. Um, Now that we show you that, we're gonna move forward to the one that was like a lot simpler to work on the left hand side. So these pigments that can give you different colors in nature composed of different kind of structures. So this porphyrin is one of the most abundant colored molecules in the nature. The reason of that is because these porphyrin core, when they're bound to um, different metal ions, they can give you this kind of chlorophyll or green-like leaves color or they can give you this kind of bloody kind of molecule in hemoglobin. This is a picture from the food that I had in Thailand, in my home country, where it's kind of bloody larb that people like to eat. I wouldn't recommend eating it if you're not used to it because it's pretty unique and it's very spicy. Um, there's some other, compounds. For example, melanins, that was like the main compounds that was the pigments in our skin, or the animal fur, and also these kind of anthocyanins compound that are found in wines or in grapes. So you see that there are some common structures or common key things in these molecules. If you don't have chemistry background, that's fine. I just want to show you that there are a lot of these double bonds where you can have these kind of like underlined line in this chemical structure. These are one of the main reasons that these compounds has the color at all because it can function in the biophysical properties so they can give you distinct color. So there are also a lot of other compounds in the nature. For example, these carotenoids compound that were found in both animal and fruits and vegetables that I want to emphasize that like these are the compounds are pretty simple and like will be repurposed in a lot of natural systems, such as the vitamin that was the key compound that benefits our our diet. So you will be informed since you were a kid that you should eat a lot of these colored vegetables because they're something that is useful for you. They're like important vitamins so that you can turn into a very functional and important compounds later on. The group of compound that I want to show you and turn us toward the color protein is this group of spatial compounds where it's composed of just a few amino acids, and it was found a lot in the coral and also like the organism that was related to the coral. So in this case, it's very unique because this kind of compound is actually composed in the protein, and the protein will just form this kind of core structure so that it gives you this kind of vibrant color instead of these, uniquely different biopigments found in the wild. So why do we care about these, spatial coral protein or coral pigments? Is because if you try to engineer these processes, um, if you're trying to make these carotenoids, for example, the astaxanthin, the red compound that was found in shrimp, and also like it was eaten by, flamingo, and then they have this pink color. It was composed of a lot of like complex metabolic pathway in order to make these biopigments. If we are, if we were to make these compounds in E. coli, for example, you have to introduce a lot of the genes. So if you count on the way here is like CRT, EB, L, Y, Z, and W, that's more than 5 genes that you have to work around in order to make E. coli colored. But if you use this compound, this protein called MUCP that came from this Acropora mellifera, from this stony coral, it can actually give you this colorful blue and purple shade of protein so that you can just have to encode just one protein to give to get this color. So that's why in our process, if we were to use this for the BioArt, it's a lot easier to just use one or a few protein encoding sequence so that we can make these kind of colors for the BioArt applications further downstream. So, um, knowing this, I want to go into a little bit of physics. I'll go very fast, but like if you're interested, we can chat further. The way that we work around these colored is dependent on the physics itself, right? If you see the leaf that is green, it doesn't mean that the leaf was making the green color. It was the way that our eyes perceive this greenness. So if you see this kind of like absorption or the reflectance spectrum, you will see that, the leaf is actually absorbing the blue and the red color, and then it just left the green and yellow part to be reflected to our eyes. So most of the leaves will appear, green or yellow, most of them. And that's based on this core structure of the chlorophyll that has the magnesium compound inside the core chlorophyll structure. If we were to go for some other different process that is not just absorption of color, The simplest one is going to be the fluorescent or phosphorescent compound, where it's just like, take the energy in form of light and then store it and also emit it back. So if you know when in your house, if you have some of these kind of like phosphorescent or fluorescent stars on your ceiling, these are one of the process where the light was absorbed and stored and then emitted to you later. So, there are a lot of like processes around here. I just added as a note. I don't want to go too far, talk here, but I wanted to introduce you to these kind of luminescence and fluorescence that was occurring in the nature. So if you go to the ocean, you see a lot of things that was emitting the light in the nighttime. And there are some processes that was required for these jellyfish or mushrooms so that they can emit and lure of the, some of the smaller organisms so that they can interact with. The first time that the fluorescence was discovered was by studying these jellyfish, where it was found to be bioluminescence. Luminescence is, the way that it just turned the chemical energy into light. And this is similar to like how you turn the light bulb on using electricity and turn it into light. But fluorescent is a different process. For fluorescent, you shine the light in, and they store the light and also emit the light back in another form. So this is light turning to light. On the left-hand side is a Jablonski diagram where it was like the way it was the physics was involved in this process. Let's skip that a little bit, but um, is actually the color was relying on the process in which it absorb and reflect, emit the actual energy in form of light. So the GFP is actually the first protein that was found on the edge of these jellyfish. So they study about it and they found that even though all these jellyfish cells can give these bioluminescence, there are some of the cells on the edge. They can give these fluorescent processes. They can give this fluorescent signal upon they were collecting the light and just emit the light afterward. So this AVGFP, was studied and can be used in a lot of processes in a lot of biological study downstream, and it was awarded the Nobel Prize in Chemistry in 2008. Osamu Shinomura was the one who discovered the GFP. Martin Chaffee was the one who used it in a lot of biological study, and Roger Xian is the one who engineered these proteins to have very different and broad colors so that we can use downstream in the biological study and also in the BioArt that we are going to use in this project. So this is an example of how you can turn these, artistic fluorescent protein to draw these, La Jolla Beach that was given in the Nobel talk from Roger Xian. And the reason that we are coming back to this kind of process is because it's a lot easier and more modular these days to work around the DNA synthesis and DNA engineering so that we can make and remake these processes much easier in a very fun way as well. You see that at the end. So the way the Green's fluorescent protein work is by having this core structure similar to what I showed in the earlier slide, and it's going to be in the middle of these beta barrel-like protein. So it looks like a barrel, right? If you see, so, how the beer making or wine making were made, this look like the barrel that was used to incubate those things. And this molecular chromophore is going to be buried inside this beta barrel-like protein. So the way that this compound is made is by having these three different amino acid side chains where they do the process called autocatalysis. So they actually undergo the chemical reaction between these, core amino acid and just turn into this core chromophore that can give you the fluorescent color, like, something similar to my shirt right here. So this is the more, um, like chemical reaction mechanism. I like this process and I want to give it here for those who like chemistry. But, the only thing I want you to take away is that these chemical structure is inside, the beta barrel. It was protected from the other molecule that may take the energy away in the other form instead of the light. So this sort of force this fluorescent protein to give away the energy in the form of light rather than, the other interaction like heat or the transfer by just, hitting each other or something like that. And the other important thing that I want to emphasize here is that most of the process to make these chromophore require oxygen as another core molecule to interact. So that give you another hint that like if you're trying to work in the system where it doesn't have oxygen, this fluorescent protein was a lot harder to work with because it doesn't have oxygen to oxidize themselves into this active chromophore form. So, when people do engineer these fluorescent proteins, most of the time it was engineered in multiple ways. The easiest approach to engineer these fluorescent proteins, chromoproteins, is to change the way the color work. So what you can see on the left-hand side here is the different fluorescent proteins where they can take the light and it is under, black light or UV light. They can just turn itself into these different cyan, green, yellow, and red fluorescent proteins. And there are also these forms of proteins that are colored but doesn't have the fluorescent. So not fluorescent, it means that it has color, but when it was shine, it doesn't return the light. So we will call these chromoproteins instead of fluorescent proteins. And they have different properties, right? Sometimes you want the light to come back and sometimes you don't want it. And the way that they behave are also different. So that's why, we're going to include the chromoproteins in our study as well. This is the way the chromoprotein sort of like, show the structure. And these are four different chromoproteins that were aligned with each other. You can see that they're very similar and they're also very similar to how the fluorescent protein that was discovered. Interestingly, these chromoproteins were discovered way later than the fluorescent protein, partly because the method that was used to study the genetics of these compounds or these proteins were very uniquely different. And it was assumed that all of these are just fluorescent proteins, but the group of chromoproteins that doesn't fluorescent just was found like later in the scientific process. So again, these are the difference at the core, chromophore of this chromoprotein. They give you different colors because they have difference in this chemical structure inside. Again, no need to dig deeper, but what I want to show here is that they're kind of different. It might be difficult for you to look further on what the difference is, but if you look at the far right here, this is the S sulfur right here, and this one is like oxygen and nitrogen in state. So those are the key difference that change the way the light were absorbed and give them very uniquely different color at the end. In terms of engineering, most of the time these core compound were used or were targeted as an engineering process so that you can turn the different fluorescent protein into different color. So if you start with a green color, you can actually make some blue or orange or red colors from these kind of core structure. So it's going to have the core beta barrel-like structure, but at the end of the day, they will give you a different colored output. So this is, some examples you can see here. You can also read some papers that was attached here to see further how it was approached to make these core different chromophore engineering. Other notes around the engineering of the fluorescent proteins is that you can also engineer the same colored protein to have different properties. And most of the time, when scientists were trying to study these fluorescent protein, they're trying to make them brighter and also more stable. So that's another thing that I want to emphasize here. In the 2025, there are also this paper that make this new M chartreuse, M juniper, and other things that are way brighter than the previous fluorescent proteins. So these are the processes still ongoing, and I'm not entirely sure, when the engineering campaign will end. But we have a lot of things that were already good to begin with, but we are also trying to make it much better and much appropriate for different usage in our workforce. And to be honest, if you were to work with this fluorescent protein at the end, they're very bright compared to the original fluorescent proteins, per protein. The chromoprotein engineering can also follow the same route where you can do a lot of engineering at the core chromophore and generate these distinct colors at the end of the day. We also have like several groups of chromoproteins in our collection so that we can use them as one of the cool approach to do the drawing that we wanted to do at the end. And Finally, there are some other engineering around, different properties. Like this is the figure from the paper. They're trying to engineer this protein for less oligomerizability. So they engineer these monomeric red fluorescent protein to have different color instead of using these native chromoprotein that tends to make inclusion bodies or tends to make a flocculation of them together. And most of the time, this kind of like big chunk of protein inside the cells is not that healthy for the bacteria. So there are also some other approach by trying to make the dimer and also the tandem dimer so that if you turn them into a dimer already, is less likely to form a dimer with the other fluorescent protein, something like that. Um, you know, in our How to Grow Almost Anything team, in the past summer and also like after the end of the previous class, we acquired a lot of these fluorescent proteins that was already available in the database and try to turn these into these kind of beautiful drawings. Um, when we found the starting point of this process, we tried to push further, and thanks to a lot of our team members, we are trying to mix the colors and also trying to tune the colors for the different properties as well. So these going to be the projects that we are going to try to work around by trying to use the diverse colors that we have already gathered in our process to make a much more beautiful artwork in the end. So these are, some examples of the artwork that was already made by our team, and you can make a lot of the different colorful artworks using the process, but you can also just try to do it by your hands. That was possible, but if you have the machine that was able to dot these proteins these E. coli or bacteria very efficiently, you can have a very different and very fine-tuned dotted works at the end. So if you're interested, this is the website that Ronan has made, and you can also upload some pictures so that you can convert it into this kind of dot art, and they will just return the file so that this kind of artwork can be used to plot in our cloud lab. It was under collaboration with Ginkgo Bioworks right away. So I think you guys should be able to try this process very readily on the website. And if there is any interest in trying to use this further in some kind of different BioArt, I think it would be a great starting point for all of us to explore further with this year of How to Grow Almost Anything. I think that's the end of my presentation, and thanks everyone for your interest in this process, and glad we can make a lot of cool artwork this year.